Hello everyone, welcome to a new edition of the Impactive webinar series. I'm Lady Flamarike, part of the Impactive Communication and Dissemination team, and I will be your presenter for today's session. So you might already know if you are here with us, but just in case, Impactive is a European project funded by the European Commission to make greener pharmaceuticals thanks to mechanochemistry. And what is mechanochemistry? This is a branch of chemistry that uses mechanical force to mix and match compounds together and then synthesizing active pharmaceutical ingredients or APIs, but without the need of solvents. So our efforts to achieve more sustainable medicines go beyond that, and that is precisely what this webinar is about. But before introducing our two wonderful speakers that are already here with me, I just wanted to say that this is already our ninth webinar. Yeah, our ninth webinar session. So in case you missed our previous webinars, you can watch all of them on our YouTube channel, our Impactive YouTube channel. So now it is a great pleasure to introduce our two speakers, Riina and Dzmitry Kananovich. So hello to both of you. And many thanks for participating today, of course. So they are also part of Impactive as they work on the sustainability strategy within our project. And they are going to introduce the challenges of assessing risk and impurities in mechanochemical synthesis. So first, Riina, she leads the supramolecular chemistry group at Tallinn University of Technology in Estonia, also known as TalTech, with Dzmitry as senior researcher in that same group. So her scientific interests include the development of mechanochemical synthetic methods, as well as the development of sustainability methods for material recycling, among many others. And she leads also the Estonian Center of Excellence in Circular Economy for strategic mineral and carbon resources. On the other hand, Dzmitry's current research interests include the development of novel mechanochemical synthetic methods in organic chemistry, particularly those involving organometallic reagents, and he began his independent academic career at the ERA Chair of Green Chemistry at TalTech. Before we start with the presentation, just a quick reminder for our audience, so you can post your questions in the chat. I don't know if it's here or here, and we will address them at the end of both presentations. So Riina, she will be presenting the first part of this webinar, and then Dzmitry will continue with a case study. So Riina, whenever you're ready, you can start sharing your presentation. Let's give you some time to prepare it.
Okay.
Okay, I can see it.
Okay. Hello. And you hear me well. Thank you for very kind introduction and welcome everyone on air. And indeed, today we will talk about Impactive, about the APIs, together with Dmitry Kananovich. So I will start and he will follow on the same topic. And a little bit of introduction. Then I talk about green metrics and assessing environmental impact and sustainability. Then about impurities, and it will be followed by more detailed case study on the Imatinib. So our TalTech supramolecular research group consists of senior researchers and PhD students all over the world, and master and bachelor students, because in Estonia, students already from the second grade are invited to research labs. So we are happy to work all together. And mechanochemistry came to our group from the collaboration with Tomislav Fričić when we looked into how to make macrocycles in solid state. And they found that dynamic covalent chemistry can happen in solid state during milling. And we also learned that it was important to have aging in order to convert around template these difficult macrocycles. And now we have already shown how to make other types of macrocycle and even to upscale the solid state synthesis to decagram with, for example, biotinyl. And together, when Dzmitry Kananovich came to our group, he is excellent chemist and well known in organometallics. So he brought in zero-valent metal activation chemistry with the Barbier reaction and the Birch reduction, and we developed very nice new methods. And not only organometallic, but also organic methods, for example, amide bond and formation and amines formation and etc. And the molecule in the middle is the Imatinib, which will be more deeply discussed today later. So our team in Impactive consists, so this is the core team, and we belong to the work package 6, which is dedicated to life cycle assessment, safety and risk assessment. And what we did, we followed the progress of the synthetic chemists in the consortia, and where possible gave advice how to design the synthetic routes, and also especially Tatjana Järg and PhD students Elina Sutula and Ketren Marlene Lotus developed analysis methods to identify impurities and to quantify them. And of course, the main difficulty was in that identification was method development, which was led by Tatjana Järg. She is using the chromatographic methods and the mass detector, the high resolution and low resolution, and even ICP-MS analysis for metals. So the most important targeted products come to our lab where their content is evaluated. And I would like to highlight Tatjana's recent work on mechanochemical reaction analysis, which was also highlighted on the cover of RCS Mechanochemistry last year. So Impactive Consortium is quite wide. We have 17 partners from different countries, and each partner has their specific role. So we are here in the middle, contributing to the risk assessment and analysis, and we do it together with the Technion, who does the life cycle assessment, and then very importantly Novartis, who is really helpful on green metric assessment of the methodologies. And as it was already introduced, Impactive is about making greener methods, how to synthesize the important pharmaceuticals. And with every chemical process, there is a use of resources, and with that, the footprint to the environment happens. It's CO2 emission or the waste which might be harmful or ecotoxic or toxic to pose a risk to people. So we are targeting those impurities which are especially difficult or important, and one should note the nitrosamines, which are especially difficult class of compounds, as they are toxic in very low doses and have been shown to be in harmful impurities in the pharmaceuticals. So how this assessment of environmental and health impact happens, and what is used is green chemistry, and also, of course, the impurities profile. So green chemistry itself is quite known, I think, already from the last century, when Paul Anastas and John Werner wrote down 12 principles, and every chemist knows them and uses in their work. So in the beginning, it just was the way of thinking about chemistry. How can we prevent waste formation? How can we minimize the toxicity by using safer synthesis, safer solvents, and design the energy efficiency catalysis is there. So the points of the green chemistry are also very relevant to mechanochemistry, as in mechanochemistry we tend to avoid solvents in the reaction mixtures. But nowadays it's not enough to just state that it's sustainable because we avoid something. There are numerical values by which we assess the sustainability or the greenness. So, and in chemistry, the first measure is the atom economy. So in ideal case, it would be that the all reagents convert to a single product, and then we get ideal value of 100%. But for complex molecules, it is often impossible. Though we can choose routes which have higher atom economy. Then the next tool how to assess the greenness is the reaction mass efficiency. How big is the mass of product per the reactants, then process mass intensity, again the mass of process per product, and the E factor, well known, the mass of waste per mass of product is widely used. And these are helping to assess also how large are the volumes of the reactors, and by that the footprint also on the organic matter needed. But one can needs to differentiate that one can measure the process mass intensity based on the reaction itself, when only materials involved in the reaction is counted, or let's take a next step when we count already the total process mass intensity and we count in also all materials that are needed for the purification and isolation. Because it is especially important for complex molecules where high purity is really needed, and pharmaceuticals are those type of molecules. So let's compare now the E factor, the amount of waste per mass of product, depending on the source of the product. If we refine oil, then it mainly means that we boil and fractionate. So the waste produced is really small because everything goes to the fractionated product. But once we rise our molecular complexity and reach really bioactive pharmaceuticals, the number of steps increases, and together with that, of course, also the waste. So it is inevitable that with the growth of complexity, also the waste increases. And if we look closer, that what contributes to this E factor, the waste, then in pharmaceutical synthesis, it's 56% of solvents. Then water, which is needed for treatment, and only 7% are reagents. So if we can somehow avoid usage of solvents, we solve several problems. One, we decrease the mass and by that become more efficient. And the second, solvents are very often also pose a risk because they are toxic, they can be ecotoxic, they can pose the risk also for chemists who are doing the work itself. So if we can minimize it, then we are in line with the green chemistry principles. And nowadays, holistic assessment is used to assess the health or these impacts, health, safety, and environmental impacts. And the simplest tool is Chem21, developed by Clark already some years ago, and it looks into the mass intensity based metrics, what I already listed, but also the risk of the chemicals, their hazards, and classifies them and helps to put the flag, whether it's a green flag, it is in the middle or it poses a risk. So we can notice it and look for improvements. So this is the first simplest way, and there are many other tools, more complex ones, EcoScale to look also the ecotoxicity, and those are developed by Merck pharmaceutical company, and IGAL, which is the innovation green aspiration level analysis, and gives the scorecards already based on the level of the technology development, whether it's an early stage or the higher stage, and allows to compare a new procedure with the medium used in the pharmaceutical industry to allow to compare whether it is new procedure is greener or not. So it is not possible to analyze sustainability without the benchmark. It is important to compare to something, because if there is no method, then the only one is the best one. So and these numerical tools are helping us in that. And of course, life cycle assessment, this is the most comprehensive approach for final process evaluation already when it's ready to be upscaled to manufacturing level. Then this is done by Technion in our consortia, and is the first assessment lab-based we can do, and Novartis helps us also with these assessments. Yes, and in order to determine better result, you need to compare it to something. So next, impurities. I talked about the health risks and where from they come. The pharmaceutical compounds are bioactive and might pose a risk to environment. But beside these pharmaceuticals, one should, the main targeted product, one should assess also impurities. Because APIs, the pharmaceuticals are to be used for the people who are in weaker health, so they are especially sensitive to the hazard. And there are strong regulations set in the pharma industry, and which impurities are looked at. There are organic impurities, inorganic impurities, and trace solvents. So the organic impurities, so where from they come, they can be just starting materials which are weren't been able to purify, intermediates, byproducts from the reaction. Also, this final API can degrade, form degradation products, etc. These are the organic compounds, because pharmaceutical ingredients, active ones, are organic compounds mainly. And it is determined that the threshold level is 0.15, and then this is the targeted that they need to be lower. So and for some, these which are really harmful, as I already noted, the nitrosamines, these are already regulated at ppm or some even at ppb levels. Then the next class impurities are inorganic, which come from the salts or metal catalysts, some residuals, and also because we are doing mechanochemistry, then the mechanical tools are usually metal-based. One should consider the contamination from the tools, from the techniques themselves. And here again, the different metals are classified according to their toxicity, depending on which amounts are allowed. And finally, the trace solvents are classified, some which are can be avoided or which are toxic, they should be avoided already in the process, so their contamination cannot happen. And for some small amounts are allowed, but then they need to be monitored. And there are some metals where there is no health risk, the macro metals, which are found in our body anyway. So, and they are easily metabolized out. So that's why their higher dosage is allowed. And where from this the impurities come, as already has been mentioned, and they need to be monitored, and they are unavoidable. The only thing what we can do, we can determine how low is their value and whether it is in agreement with the regulation. Where from they come, of course, from the synthetic step, from the catalysis equipment, solvents. And even though in mechanochemistry we tend to say that we do not use solvents, we do in the experiments in solid state, actually in many mechanochemical reaction, liquid-assisted grinding is used. So a little bit of liquid is needed for the reaction to happen, and that's how the solvent traces end up in the reaction mixture. And after the reaction, there is a purification procedure. So again, these liquids, solvent traces can come from that. And of course, starting material and reagents are the ones. So in IMPACTIVE, we constantly monitor the target APIs purities in our lab, and this is one nice highlight on metal content in one API which was studied, and it is compared to the daily allowed dose for the determined by the European Pharmacopoeia. So, and one can see that daily dose and concentration limits is lower than daily dose. And if you compare the numbers in the columns on the right, then you can see that all these metals were really below the limit dose. So we checked how the jar material can pose a risk for contamination, and found that in these cases the risk was negligible. So at least these type of vessels are safe to use. And one should mention that this didn't actually happen by accident. The route of the synthesis was also planned so that the metal impurity contamination was minimized by choosing already reagents which pose lower risk, and then also the reaction conditions. So that's what chemists can do. And further on, how our workflow goes in IMPACTIVE. So first, one needs to identify and quantify the compound, what we are looking for, targeted compound, and we do it by assay. And the final compounds products are expected to be over 95% assay value by weight, and this is the optimization target. Then we look to organic impurities, identify those which are higher and propose the mitigation measures, and then trace metals, as I already showed, and genotoxic impurities. And these ones are the nitrosamines, which we can measure in our lab and looking at their levels. So yes, once these impurities or the additives are identified, then you can search for investigation where from they come, and after that, of course, the mitigation strategy, how to purify or exchange some ingredient. So it is really important to note that best mitigation strategy is, of course, prevention. If one is not using can avoid some toxic intermediate, reagent, or catalyst, then this is the way how to prevent the contamination. And from this on, I would like to give a hand to Dmitry Kananovich, excellent chemist, to talk about synthesis of Imatinib. So, yes, thank you. And I will stop sharing my screen, and Dima, you can take over the floor.
Thank you, Riina, for your presentation.
So I'm going to continue now.
We can see it.
Okay, great. Then let's continue. So hello, everyone. So I'd like just briefly present a case study for Imatinib synthesis to illustrate what was said by Riina. And first of all, we have to confess, of course, that Imatinib, in fact, is not one of the IMPACTIVE target molecules, because we started this work before start of the IMPACTIVE project, but it was completed at right time in the beginning of the IMPACTIVE. So this work offered us a very convenient opportunity to elaborate and validate the workflow that we currently use in IMPACTIVE in general. So regarding Imatinib itself, this is a molecule with a prominent history. It was developed actually by our partner Novartis as one of the first rationally designed anti-cancer drugs. And at some point, it became a bestseller. So with sales, as you can see, reaching billions annually under a trade name of Glivec. Currently, it is also included into World Health Organization list of essential medicines. And the most common, though not only existing retrosynthetic disconnection reveals three key building blocks here, namely heteroaromatic amine 2, 4-hydroxymethylbenzoic acid 1, and then methylpiperazine, which can be assembled to build this molecule via carbon-nitrogen bond forming reaction, like amide coupling and nucleophilic substitution and benzylic position. And luckily, both of these methods are very well developed in mechanochemistry. That's why this target molecule is so good for us. And I will show you today a simplified synthetic route and not complete synthetic route, as we don't deal here with preparation of this heterocyclic amine 2, which is quite complex already by itself, but luckily it's commercially available. And to begin, let's first look how this molecule is made under conventional solid-base synthesis. Actually, there are many patents and publications available. I just selected a single representative kilogram scale route shown here. And so it starts with activation of 4-hydroxymethylbenzoic acid to substitute hydroxyl groups with chlorine atoms and prepare this acid chloride. Then amide bond formation step in chloroform, and finally nucleophilic substitution reaction in the methylformamide. And if we apply green chemistry toolkits to this route, for example, Chem21 signalize us that yields are maybe not so good here at both of the steps, and also this route involves hazardous chemicals and much more important, hazardous solvents. Another problem here, there are two genotoxic intermediates. The starting material, amine 2, is genotoxic, and the intermediate, chlorinated intermediate 4, is also genotoxic, and both are regulated by European pharmacopoeia. You can see the maximum thresholds are in ppm range. And so our idea was basically to eliminate hazardous solvents by using mechanochemistry and avoid toxic reagents, and at least to bypass one genotoxic intermediate, as we hoped alcohol 5 intermediate here should be less dangerous, considering perhaps DNA alkylation as a placeable mechanism of genotoxicity for chlorinated intermediate 4. And to make the story short, here I show our final optimized mechanochemical route. This work has been published a couple of years ago in RSC Mechanochemistry. Okay, and basically the first step is very well developed in mechanochemistry, carbazimide mediated amide coupling with ethyl acetate as a liquid assisting grinding agent that delivers a high 94% yield of intermediate 5. And then the next step basically involves activation of a benzylic hydroxyl in intermediate 5 towards nucleophilic substitution with amine. This chemistry was developed in our lab previously by Tatiana Kananovich, who was also the main person behind the synthesis as well. And this step delivers Imatinib, 96% yield. And from a practical standpoint, that's how synthesis looks like. So basically, we perform both steps in a shaker mill using zirconia-coated milling jars with a single zirconia milling ball. You can see here the first step, but second step looks essentially similarly the same way. So you can see solid reactants placed in the milling jars. That's the reaction mixture after the milling. And the workup is pretty straightforward in the both steps, just treatment with water, infiltration of the solid product. And purification involves just recrystallization from methanol ethyl acetate. Okay, and now it's right time to talk about impurities. And in this work, I would like to highlight we focused only on the identification and quantification of minor organic impurities and genotoxic intermediates. Here you can see HPLC chromatogram of intermediate 5, after so product after amide coupling step, where we have high 98% HPLC content of this compound, but 2 impurities are present. The most concerning one is genotoxic amine 2, which was a starting material, and additionally we identified doubly acylated ester impurity A as another byproduct. But this luckily can be eliminated by avoiding either excess of hydroxy methylbenzoic acid or alternatively by treatment of crude product with just aqueous alkali instead of water, which converts A into intermediate 5, which is the best strategy in this case. And for the second step, Imatinib synthesis, we identified additional impurities. So in this case, impurity B, which is a doubly alkylated piperazine, and impurity C, which basically derives from impurity A, as it clears from its structure. Luckily, Imatinib can be essentially purified by simple recrystallization. So recrystallization step leaves only trace impurity C and a little bit of intermediate 5. But the much more important question, whether these impurities are dangerous or not, are they genotoxic? With these questions, actually, this question we asked our collaborator at Novartis, Dr. Fabrice Galou, and who performed for us a standard for pharma industry in silico assessment for all molecular entities in the synthesis in order to understand whether these might be genotoxic or not. And to our delight, he returned us with an answer that the only dangerous molecule was amine 2, but not all these other impurities and intermediate compounds. That it is a good sign for us. And therefore, we turned next our attention to the impurity of amine 2, which was first quantified in group Imatinib, and we have quite a huge concentration, above 500 ppms. so well above the threshold limit, of course. And if you can recall, we had quite a lot of these impurity already after the first step. So what we did, we modified the experiment protocol for the first step and workup protocol to increase assay of this crude amine and to eliminate this trace impurity of genotoxic amine here. And it was successful. So we got much more pure intermediate, and this refinement basically reduced content of 2 in Imatinib to 86 ppm already. So still it's quite a lot, but it is comparable with content during obtained during conventional synthesis in solution. And there are protocols available how to purify the product further and remove it below the threshold level. So that's why we stopped at this point, the development. Now let's turn our attention to green chemistry metrics. First of all, we assessed our new method with innovative green aspiration level approach, which is specifically tailored for pharmaceutical synthesis. And our early development approach showed excellent relative process greenness above average industrial benchmark, I can say. And now if you compare some green chemistry metrics of our route versus solvent-based route, so we can see that, we really introduced some improvements. So first of all, the total yield is much higher. And we have, well, quite comparable PMI values, the total PMI values, even despite our method is just early development stage, not kilogram scale or something. And usually PMI decreases when you upscale your reaction. And this is due to mostly, due to better optimized mechanochemical conditions. And of course, we managed to replace toxic solvents like dimethylformamide in chloroform with just environmentally benign like additives, ethyl acetate, water, which is used for workup, and dimethyl isosorbate, which is bio-based solvent. So, but does it mean that our method now is perfect and we have to stop at this stage? Actually, another very big and great advantage of green chemistry metrics is they showed points where further improvements are necessary, basically disadvantages. And we can notice that still atom economy is quite, I would say, low for our method because we utilize chemicals with high molecular weight. So atom economy can be improved by selecting maybe alternative reagents. Same about reaction mass efficiency. This is also quite low here because we use as a second step excess 10 equivalents of N-methylpiperazine. It also can be improved. And another thing, of course, even though reaction PMI is low due to avoidance of solvent, we still use solvents to, like water, mostly water, of course, to purify our product into isolated and pure form. And this highlight one of the well-known issue in organic mechanochemical synthesis with community have actually to solve, that we still have to optimize our workup protocols to really reduce PMI also here, not only on the reaction stage. And of course, genotoxic compound, the still disadvantage of this method, we still, as we already discussed, the best method is prevention. We still rely on this genotoxic starting material, so a better synthetic route should avoid it. But this means that this retrosynthetic disconnection need to be revised a little bit. Okay. And based on this case study, actually, we developed our current impactive process development workflow, which is shown here on this chart. And synthetic protocols which are developed by our colleagues are being routinely analyzed by green chemistry metrics to mitigate all issues already at very early stages. And simultaneously, here we analyze also samples of all active pharmaceutical ingredients produced to check them for impurity levels. And our colleagues at Novartis assess their hazardous potential, similarly like I showed before. And based on this, we constantly refine our synthetic route and select the leading one, which is good from the point of view of green chemistry, sustainability, and health issues. And to conclude the whole story, what was said by Riina and by me, if I may, I would like to leave maybe two take-home messages. First of all, that one shouldn't really hesitate to present disadvantages in green chemistry metrics analysis. These are also very important, and they highlight problems that need to be solved and open new research directions. And second, we showed many times that prevention is the best mitigation strategy. Basically, it is extended version of first principle of green chemistry. So better to avoid problems and later deal with the problem. And we try to adhere to this principle in IMPACTIVE whenever possible, and it shows quite good results. So thank you for your attention. That's it.
Thank you so much, Dzmitry, for a very nice presentation. And thank you again to Riina for presenting the first part. So now we have still a little bit of time to address some questions. I think there's already one from our audience, so let's address that one first. So this is related to the Imatinib use case. So we have someone that is asking, but you noted some anhydride byproducts that can still form. So can we control the anhydride formation, particularly during a scale-up?
As far as, yes, for some reason, I have a little technical difficulty. I see nothing on my screen, so sorry for that, but I can, of course, answer this question. So the byproduct that I showed, actually, it was ester, not anhydride. But yes, in some cases, indeed, as we worked quite a lot in mechanochemical amide coupling reactions, indeed, anhydride can form as a byproduct as well. Yeah. For these, we need to properly select reaction conditions, and there are some conditions which allow to avoid this. So it seems to me we have a kind of small study even presented in this paper published in the CEST mechanochemistry. So I recommend to have a look there, maybe.
Perfect, thank you. And let me check if there are any more questions. If not, I have some of them prepared already. So this is a new one that we noticed in some of your slides that some impurities, I think this was a Riina slide, need to be detected as low level as ppb, so parts per billion level. So this is a super tiny, small amount. So how do you do that? How can you do that? If you can tell us a little bit about the techniques you are using here.
Yeah. So yes. So these specific low amounts can be detected by mass spectrometry. We need mass spectrometry for that, and different types for depends if it's a metals. Then it's this ICP, which evaporates in plasma flame all the organics and detects specifically only metals. If it's organic compounds, then we go to very sensitive high resolution and sensitive detectors, which are separately need to be calibrated in order to achieve these low amounts. Yes, so these low amounts need special methods, calibration methods, and to achieve a standard. So yes, it's very time consuming to do it.
I can imagine that. Yeah, and there are super tiny amounts. Okay, perfect. Thank you, Riina. And we have a follow-up question from the same person. So he's saying, since solvents are still needed for the work purification strategies. What
Exactly.
are the best green strategies to purify APIs without raising the overall PMA again?
Yes, that's exactly
I
the
don't.
thing. That's exactly the
Okay.
thing, which is being actually discussed now in the mechanochemical community, how we can also innovate this workup stage. So they can really avoid also solvents at the work type stage. So this area is open for innovations. I can only say you that at the moment, what I showed you, perhaps this treatment with water is the best method. So just use water and that's it. Yeah, and probably we can minimize. Okay, it was early stage development, early stage demonstrated. Probably we can still minimize use of water here. But water, even though it's a green solvent and has its own disadvantage, then you have to deal with remediation of water afterwards.
Of course, yeah.
So sometimes even organic solvents are better in this regard. So, but this is one of the bottlenecks, current bottlenecks that you have really to address as a community. I mean, mechanochemical community.
And I see Riina noting, so maybe, yeah, please, Riina, go.
So maybe I can add, yes. So the purification, if the reaction is, of course, the giving highest purity and everything is consumed to the product, which is usually not the case for complex molecules. So the purification step must be there. But now the question becomes, if you compare solution-based or mechanochemical synthesis, also in solution-based synthesis, purification step is there. And usually some properties of the molecule, if it's already known how to purify it. So it can be just taken over, or now one can think in which form in solid state this final product is ending, that its purification is easier. So this is about the innovation that Dima were talking about. But I want to add to this discussion about the impurities and mechanochemical reactions that actually as mechanochemical reactions are reacting solids, then these are really sensitive, these reactions, to the small solvent traces, impurities, which came in the starting material. So in solution-based, if the solid is dissolved in the bulk solvent, then small traces, they don't matter because statistically they are really negligible. But in solid state, they become a stoichiometric, they might become a stoichiometric reagent. So in sometimes the small amounts of additives which are neglected in solution phase cannot be neglected in the mechanochemical reactions.
Actually, you mentioned that during your presentation, if I recall correctly. Yeah, and I had precisely a question a little bit related with this one, which is that I assume that assessing impurities is key in chemical synthesis, but it's even more important if we are talking about API synthesis. So here is really, really important as we are talking about APIs plus solid state synthesis. So I imagine that this is adding an extra layer of difficulty here. So how are you dealing with that?
Yeah, absolutely.
Who wants to answer?
Yes, because as we already mentioned many times, yes, we have to quantify some impurities at part per billion level, and this is extremely time-consuming and challenging task. So yeah, so it's absolutely another level of difficulty. And yeah, even talking about generally, like maybe more fundamentally, maybe even didn't study it well still how impurities are formed under mechanochemical conditions. So how the reaction is, how to say two impurities sometimes can react, yeah, and produce certain impurity, yeah. So kind of mechanochemistry under highly diluted conditions. So we fundamentally probably know almost nothing about it. So it's by itself, it's quite an interesting research area.
Yes, and whether it is made by mechanochemistry or another way, so APIs have regulations and the standards need to be met. So this is the level. But yeah, the research is in there that to understand how reaction proceed in solid state, and so this is really needed.
Yeah, because it's a kind of a new field, let's say like that. So we need to really do some research here to discover what is going on within the reaction, which we have work packets focused on that. So yeah, that's important to take into account. And another question from the audience: can turning the liquid assisted grinding additive completely suppress this anhydride byproduct? What will be your views?
Could you
Riina,
please
you're smiling.
suppress?
Suppress this the byproduct we were mentioning and writing
okay. Yes.
the question here on the chat that we have here.
I can maybe say that we observed up to now opposite. Yeah, but maybe yes, sometimes it can suppress. Yes, so yeah, absolutely. So selection of lac additive is important. That's what we learned also from the project already. So hopefully soon some examples will be published. You will see it.
We hope so. Yeah. Okay. So
and it depends on the target. It depends. I don't know whether the person who asked was talking about Imatinib anhydride again or some other, but it depends on the specific reaction and this field of the polarity influence of the solvent additives and also other additives. So this is this in mechanochemistry is really demanding, and even amount is the same solvent, but in the difference to geometric amounts can strongly influence the outcome. So there is many layers of the mechanistic pathways.
For mentioning Imatinib, because I recall now. So that's how we selected lac additives in that work. Seems to me we have a table in the supporting information, and that indeed different lac additives producing different level of impurities. Yeah.
Okay. And we have talked about impurities, but I would like to ask impurities generated during synthesis, but does your work also include ideas or solutions related to medicine disposal? As this has been mentioned during the webinar that we are talking about bioactive compounds, so they might have an extra risk for the environment where if we just dispose them freely.
Yeah, this is the big problem because the dosage people consume medicine are needed and they have their shelf time, and one topic is what to do with the non-used or disposed medicines, and there are some groups even looking into how to reuse these complex molecules. Definitely bioactive molecules pose hazard. They can degrade in environment if they are not used, and some of it we do not control. If we consume, then they come out of our body also and the problem of the pharmaceuticals, it's not only the non-used one but also metabolized and growing problem. And one direction of studies goes with the looking how these bioactive compounds, pharmaceuticals, can be degraded or in the environment in safe ways, but before that they need to be sensed also. So there is different steps before you reach this degradation. If they are in your hand, then it is easy already to work with them. But if they are diluted in the waste, then it is posing the problem. So this is absolutely difficult field which needs a lot attention, and constantly the sea and our nature is now monitored by the environmental chemists, looking at how the traces of antibiotics and are influence our own nature.
Yeah, antibiotics are a huge problem now, antibiotic resistance. Yeah, and Dzmitry, sorry, go, please.
Oh,
No,
basically,
I thought you wanted to add something.
I think Riina answered it well, but yes.
Okay.
But on the other hand, yes, I can only add that yes, what you can do definitely if you have something in hands, I mean, just to dispose undiluted pharmaceutical or something, yes, that of course we can develop perhaps some mechanochemical degradation pathways. I think it's not a big problem, could maybe, but yes, it's another thing when it's in environment, yeah. So in this case, the best strategy is benign by design pharmaceutical, but basically
Yeah.
it requires, collaboration of different disciplines, basically pharmaceutical chemists who develop drugs and chemists who study biodegradation and so on. So yeah, it's not so easy.
Okay, so thank you so much. We need to cut here as we've been here already 1 hour. It was a really, really interesting presentation by both of you. So it's been a pleasure to have you with us and to learn more about risk and impurity assessment. Just one last thing. To our audience, if you want to stay tuned to our latest updates, you can subscribe to our newsletter. Also, you can follow us on our social media channels. So we are on LinkedIn and X. And of course, we have a webpage, which is mechanochemistry.eu, where we also post our latest news. And well, thanks again to you both, to Riina, Dzmitry, for participating today, and to everyone at home for joining the webinar. And of course, thanks also to the European Commission for funding the project. So we hope to see you soon on our next session. Okay, thank you. Bye.